Hi everyone, today we will be looking at how thyroid hormones are synthesized and secreted by the thyroid gland. So I thought we would start out by reminding ourselves of what the thyroid gland looks like and where it's located. So on the upper left you can see that the thyroid gland is a butterfly-shaped gland located just beneath the thyroid cartilage. And here is the laryngeal prominence, otherwise known as the Adam's apple, just so you get an idea of where it's sitting in your neck. And usually the thyroid gland will span from the C5 to T1 vertebrae. Okay, now we're going to zoom in and have a closer look at what the thyroid gland looks like. So the functional unit of the thyroid gland is the thyroid follicles. So these are the big circles you can see everywhere. So the thyroid follicle is formed by follicular epithelial cells that surround it. This cluster of cells you can see in between the follicles are known as parafollicular cells or C cells, and these are involved in calcium metabolism, but we won't be talking about them today. Inside the thyroid follicles is colloid, which is the protein precursor to your thyroid hormones. Okay, now we're going to take an even closer look at a single follicular cell. Let's zoom in. Okay, just to orient ourselves, on the left is the blood, so this would be obviously inside capillaries. In the middle we have our thyroid follicular cell, and on the right we have our colloid. So thyroid hormones are made from two substances, iodine and the amino acid tyrosine. Our bodies naturally make tyrosine, but we need to get our iodine from our diet, which is why we consume iodine-rich foods like fortified table salt and bread. Iodine, which is I2, is absorbed and digested in the form of iodide, which is I-. Okay, so where is tyrosine used? So the endoplasmic reticulum of the follicular cell produces a protein called thyroglobulin from the tyrosine amino acids. This is exported from the cell by exocytosis to become the colloid. So here it is in green. Okay, so I said before that we also needed iodine, so how are we going to get that to the colloid? Well, it enters via the sodium iodide symporter, which is a form of secondary active transport. It then diffuses passively through the protein transporter called pendrin. Before it can combine with the thyroglobulin, it needs to be oxidized or activated by an enzyme. The enzyme is called thyroperoxidase, or TPO for short. Here it's in pink. So our iodide has now been oxidized to iodine, I2, and it's now ready to combine with the thyroglobulin. This process is known as organification and is catalyzed by the same enzyme, TPO. These are the tyrosine amino acids to which the iodine molecules will be added. If only one iodine molecule is added, it is called monoiodotyrosine, or MIT for short. If two iodine molecules are added, it is known as diiodotyrosine, or DIT. Okay, we're almost there. Just as a reminder, the two thyroid hormones are triiodothyronine, also known as T3, and thyroxine, which is T4. One MIT and one DIT molecule are combined to form T3, and two DIT molecules form T4. This coupling reaction is also oxidized by TPO. Here at the bottom of the thyroglobulin molecule, you can see what T3 and T4 look like. Okay, so now we have our thyroid hormones. How do they get into the bloodstream? The thyroglobulin is endocytosed by the follicular cell. The vesicle fuses with the lysosome, and the thyroglobulin is cleaved to release T3 and T4. These are now free to enter the bloodstream. T3 accounts for about 10% of the thyroid hormones that are produced, and T4 the other 90%. And that's it. I hope you now have a better understanding of how thyroid hormones are created and secreted by the thyroid gland. Thank you.